So it's a great pleasure for me to introduce my colleague, Ron Shamir, who is a professor of bioinformatics at the Blavatnik School of Computer Science at Tel Aviv University, and also the head of the Safra Center for Bioinformatics at Tel Aviv University. Ron is highly recognized for his contribution to the development of algorithms and tools for understanding the genome and human disease. He received many prizes and awards, including the Kadar Prize, the Landau Prize, and Test of Time Award for his, I think, 2 Recomp papers. He's an ACM fellow and also an ISCB fellow. Thank you, Ron.
And thanks for the invitation. So I'd like to start with a very high-level description of the kind of world that I'm working in, and then zoom in on 3 projects with not many technical details. So this is statistics of the 10 best-selling drugs in the US. And in blue, you see a person that was helped by the drug, and in red, you see the proportion of persons helped by the drug. So heartburn, 1 in 30, asthma, 1 in, I don't know, 25. And these are the best-selling drugs that are available. So even the best drug that we have fail much more than they succeed. And this is even worse. Look at statins. Statins are the killer drug for, well, blockbuster, I should say, drug to deal with heart disease. You need to treat 104 persons in order that one will be helped to avoid the heart attack. You need to help to treat 150 so that one prevents strokes. On the other hand, 1 in 10 will be harmed as a result of muscle damage. And these are not exceptional. These are actually the best drugs that we have. And the problem is, of course, that we are using one-size-fits-all. Each of us is different, and we are using the same drugs, the same treatments for all of them, without really tailoring this to the features and to the genetics of the individual person. So the goal is to identify precise medication and treatment and to tailor it to the individual. On one hand, there is a huge opportunity because of the big biological data of the type that Aviv was telling us. So by now, there are dozens or maybe hundreds of assays that can be applied to an individual, starting from his or her DNA, the expression of the genes that Aviv talked about, the levels of the proteins, metabolites, and more. So each of these, you can think of it as a vector of lengths a few thousands to a few millions. And we call it with a nickname omics. Each of these, they are transcriptomics, genomics, etc. So generally, we call them omics. And one can compute, one can assay them relatively cheaply by now. And actually, it can be done not just once per individual, like the DNA, but you can do it over time in the development of a disease. You could do it at different tissues and on single cell, like Aviv was describing. So this is really a great opportunity, but also a great challenge. The assessment is that by 2025, DNA sequencing alone, which is just one of these techniques, will exceed 10 to the power 21 bases. So we really are talking here about very, very big data that we can use to help us in precision medicine.
So what is the
Yes, yes.
Across all of humanity.
Oh, humanity and experiments and multiple repetitions, etc. Yeah. So on the other hand, there are the electronic medical record that can also assist us. So these are the records that are collected by hospitals, HMOs, and all other health-related organizations. And they collect the information about us when we go to the doctor or an operation, etc. So some of this data is structured, like lab tests, billing codes, which are actually identification of the disease, drugs, etc. Part of it is free text written by the doctor, and part of it is images or scan data, like MRIs or X-rays or scan text documents. So, and they are available in those health management organizations. So the vision is to analyze together the medical records of an individual together with the omics that we can collect for him in case of disease in order to achieve the precision medicine. But in order to be able to understand what does the combination of the history and the omics means, we first have to do this on a very large scale on millions of individuals in order to understand the relevant medicine and biology. So to look at very large cohort, very large groups of individuals and analyze together their medical records and their omics data. Now, so we are talking here about two types of data. One type is the biological or omic data, and luckily the genomic community has been amazingly successful in making all data that is created available for research to everyone. So you cannot publish a paper in this field without releasing the data. And there are fantastic databases that people have been using and analyzing all the time. On the other hand, the clinical data is extremely siloed. It's kept within the organization, and it's almost completely inaccessible to researchers. And in order to complete the vision that we are talking about, these silos will need to be broken down, or at least relaxed so that the data could be used. Because right now each organization only has access to its data. So the challenges are multiple. First of all, there are challenges on the level of the society. As I said, to make the clinical data available. There are, of course, issues of privacy and security. There are technical aspects. It's huge data, so the volume, the speed of computation is an issue. The data comes on many scales, many types, funny distributions, and many biases, and the statistics are complex. There are algorithms that need to be developed. This is where mainly my research focuses. And of course, hopefully once we have done all this, this has to be translated to the clinic, to be used to help the patient. And this means immersing this into the health organization. So this is the situation that we are now. On one hand, a great opportunity, great resources of data. On the other hand, accessibility and integration of these data are a big challenge. So I'll talk very briefly about 3 projects. Baby project toward this huge goal that we've been doing in my lab. And the first was done by Gal, a graduate student in the lab, together with Didi, who is a previous student, who is now doing a postdoc with UNN Ashley and Eric Ingelsson at Stanford, so they were also on board for this project. And the project deals with hypertension. So about 1 billion individuals in the world suffer from hypertension, and this is manifested mainly by high systolic blood pressure that leads to many heart problems, strokes, coronary events, etc. And there are many, many drugs to treat high blood pressure, and the normal indication is if SBP is above 140, then you medicate the patient, and you increase the medication if the level keeps staying above this. And this does help to reduce the risk of cardiovascular events. And the hypothesis of a study, the big study, a sprint study that I'll be talking about, is that maybe we should go down from 140, when maybe we should treat people with 130, 120. So this was a study, very big study of nearly 9,000 patients, where they were randomly split into 2 groups. One was given the standard treatment, and the other was given an aggressive treatment, namely increasing the medication every time the level was above 120, and the patients came back to the clinic every 3 months, and depending on the treatment arm, they were given additional medication in order to follow their blood pressure. And actually the study was so successful that it was cut in the middle. So they didn't, even before the end, they could realize that the people who were on the aggressive regime had fewer heart attacks and fewer cardiovascular events. So the technical term is hazard ratio. So the people in the aggressive treatment had only 3/4 of the events that people in the standard treatment had. And actually, as a result of this study, the indications of the American Heart Associations were changed. So the treatment is now changed. However, there is a downside. If you see, the people with the aggressive treatment had 3.5 more chance to develop kidney problems. And these are, this is a serious caveat. And what we set out to ask is, can we predict an individual's outcome better? So we looked at the data that was available from the study. Most of it was demographic information and heart, blood pressure, etc., collected before the study began. But there were also measurements taken every 3 months. And we decided that we want to look at them as well because we thought they are indicative. So we took a window of 6 or 12 months before the event, and we ignored the measurements here, but we added the measurements that were earlier as additional features to the model, and I won't go into the details of the machine learning tool that we used, but the point is that we used both the pre-study parameters and the longitudinal measurement taken during the study, and we did this both for the cardiovascular risk and for the kidney events. So here are the results for the cardiovascular events. So there were several studies that did this before. Actually including a group of the Clalit who was the winner in the study of the New England. But you see that by using the longitudinal data, we are getting far higher in terms of area under the area under the curve, the ROC curve. And we also did this on an additional study, an independent study of about the same size. We applied our predictor there, and again, we see the same result, that we do get improvement by using the longitudinal data. Now, and we got similar results for the kidney. And actually, here is a picture of the predictors result for the heart outcome and the kidney disease risk. So higher is more risky. And you see in red are those patients that, so each patient was predicted for both risk by our model. And you can see in red those that developed cardiovascular disease, and in blue, those that developed kidney events. And you see that the red points tend to indeed to be to the right, and the blue points tend to be higher up. And we reasons that maybe we can predict both of them together and balance the risk between the cardiovascular event and the kidney event. And I won't get into the details, but by combining the two predictors and using logistic regression, we ended up coming up with a recommendation system that indeed for the group that had the intensive treatment, the hazard ratio for cardiovascular event is higher, but unlike the original study, the risk for a kidney event was lower. So one can combine predict both risks, and depending on the relative weight that the doctor may be giving to them, one can choose how to treat the patient. I should mention that this part did not use the longitudinal data. Of course, this has to be validated on an additional data set. So moving on, I'll talk about the project with Nimrod Rapaport, a PhD student in the lab, that deals with integration of data of many types. So one of the open silos, I would say, that has been tremendously effective in the community is the Cancer Genome Atlas. It's an international project centered in the US that looked at many thousands of cancer patients. Here you see 12 types, but by now they have more than 30 cancer types. And for each cancer, they collected a set of patients, a few hundreds, up to 1,000, and they assayed many omics, so genetics, gene expression, etc., and the clinical data. So this is, ideally we would want all the data to be in that phase, but at least this data we could analyze. And it's not so easy to analyze because each of these omic types has a very different distribution, it has its own behavior, but we wanted to see if we can analyze them together. So can we, so each of those cancer types, like breast cancer or colon cancer, is actually a combination of several subtypes, and each subtypes may need different treatment. So if we can identify the subtypes better using all these data, maybe we can come up eventually with better treatment. So the idea was to cluster the data, but to cluster the multiple omics types together, not including the clinical data. And there were quite a few methods available in the literature to do this. And we looked at a few of them. The leading one according to what we could identify, and actually there was quite a few methods that were that are called multi-view methods that were developed in the machine learning community and never used on genomic data. And we set out to benchmark them. So we took 10 types of cancer. For each one we had a couple of hundred up to 600 patients. And we looked at 3 omics types that we understood best, and we thought they could be used together. And the clinical information that was available, so like the cancer subtypes according to what the doctors identified, and also survival. We know what happened to the patient later is something that we used for the evaluation. And how do we evaluate this? We create a group of patients based off our clustering, and then each group of patients, we look at the statistics of survival, and if the groups are indeed separate subtypes, we expect that the survival plots will be well separated from each other. So I'll show you the result of the benchmark in a minute, but by doing this benchmark, we realized that we learned a few insights. First of all, there were some pretty good methods that were using similarity data, and actually they only looked at k nearest neighbors for k equal a few dozens. So here we are clustering the patients. Yeah, we have a set of, say, 500 patients, and we want to cluster them based on similarity of their omic profiles. So we thought maybe this is the crux of these methods, and we don't need all the additional layer. And also we observed that sometimes if we split large clusters, we get better results. So we took these two simple insights and we developed our own algorithm, which we call NeMo. And it's extremely simple. So we compute sample similarities. We do this in every omic separately. And in every omic separately, we look at the k nearest neighbor of each patient sample and average this information in a weighted fashion. I won't get into the technical details. And then we just sum the similarities across the different omics, the simplest way possible, no weights or anything. And then we have a matrix of similarity between all pairs of patients, and we perform spectral clustering, a classical way to partition the set. It's a relaxation of the normalized cut method, to those who are familiar with it, and it's using the graph Laplacian. And to determine the number of clusters, we took the idea of the eigengap, which looks at the difference between the values of consecutive eigenvalues. But we did a twist and multiplied it by I, the index of the eigenvalue. As I said, we wanted to nudge the number of clusters slightly higher. So there are actually one parameter here, which is this k, and we choose k based either or the known number of clusters in case we know it. If not, we just guess it. And one neat thing about this method is that it can directly address missing data. You don't need imputation. I won't get into the details, but this is something that is easily done here. In terms of complexity, if we have n patients and p features, so the total length of the vectors of all the omics, then it's quadratic in the number of patients and linear in p to compute the similarities. This would be required by every similarity-based method. And the spectral clustering is cubic, and we do it only once, as I said. The other methods do this iteratively. Okay, time for the results. So what you see here are the 10 algorithms that we tried. The last column is NeMo. And here we count how many of the 10 cancer data set the algorithm obtains significantly different survival plots. So as I say, we look at the survival plots of each group of patients in each cluster, and we see that NeMo is first. If we look at other clinical parameters, we see that there were 4 winners, including NeMo, in terms of the highest number of data set that had such significant parameters. In terms of the number of clusters, you see that some methods do well with a large number of clusters, while others do well with a low. We are sort of in the middle. In terms of runtime, we are the second fastest after the spectral clustering, and far lower than many of the other methods. So we have here an algorithm that is better in terms of partitioning the data, and also quite fast. Here is another way to look at the result. Here you see the minimum, the mean minus log probability of separation between the set, and the further we are to the right, the better. And here is the minimum, an average number of significant clinical parameters, so the higher the better. And we see that there is no one ultimate winner, but NEMO is second in both parameters and is on the Pareto optimal contour. So one of the follow-ups that we did, we looked at one specific type of cancer, of leukemia, AML. So this was nearly 200 patients. And again, we looked at the 3 omic types. Not all the patients had the full data. And the result, you see here the 4 clusters. So here you see the survival as a function of time. So this is the fraction of patients that survived after X years. So you see that there is one cluster of very high, very good survival compared to the others. And NEMO was indeed best in terms of separating the clusters compared to the other algorithms. It was also better than using all the full data on the subset or on using any single omics, and it also matched well to known classification of AML that is available in the oncology community. We did a follow-up test that was very interesting, and we looked at the mutations in the patient's data within each cluster. So we had the 5 clusters, each one is a separate color. And what you see here is the rate of mutations of the 6 most frequent mutated genes. So I should emphasize that this data was not used as part of the clustering. It was not used to build the omic. And actually, Liran Shlush, our oncologist collaborator on this project, was very enthusiastic about the result because we did identify some of the hallmarks of AML in separate clusters. And in fact, he directed us to another data set of, it's called BeatAML, a project that currently has some 400 patients, separate data set, independent from ours. And here you see, using only the expression data in our clusters, we separated the patients in this other study. So you see here that in 2 colors, the 2 studies, and they really match very well. And you see here that the coloring does extend to this other data set, and this could allow us to enrich the set of mutations in each of the clusters, and some of them are now tested experimentally in Liran's lab. Finally, the last project is also a work of Gal, and here we are looking at trying to identify cancer driver genes in an individual. So, say again?
Who else was on that picture?
It will show up again. It's a prodigy because the project is called the Prodigy. Yeah. So in every typical cancer tissue, there are dozens, sometimes hundreds of mutations, but only a few of them are important in the sense that a change in them drives the cell into a more aggressive situation. So you see this mutation, the first driver mutation occurs in a normal tissue, and then it causes a proliferation. And these cells, like all other cells, keep accumulating mutation. And at some point, another driver gene is mutated, and the full-fledged cancer and perhaps metastasis are formed. So there is a chain of driver mutations, typically 3 to 6, 7. out of maybe a couple of hundreds that could be identified in the patient. And there are many studies that have been trying to identify driver genes because they are the basis of many of the drugs that we have, but it's typically done on a cohort, on the group of patients, all patients that have this type of cancer. And there's actually even a database that we'll be using that validated drivers. And our goal was to come up with an algorithm that can identify drivers in an individual. So why is this needed? So here you see the number of mutations, number of mutated genes, number of patients with this number of mutated genes in a lung cancer cohort. And you see that some patients have 20 or more mutated genes. Some, according to the database, some patients have none. And if we want to treat the patients, we want to prioritize a few genes. We can't give 10 drugs. It will kill the patients. It's also, no HMO will cover it. It's extremely expensive. So if we want to use combination therapy, we need to rank a few top most likely drivers in the patients. So there were previous approaches that tried to do this, two algorithms, DonRank and SCS, and they both combine the information on the mutation and the expression with a protein-protein interaction network that is available, collected in the biological community. The interactions between genes give us a clue of how one affects another, similar to the way that Aviv was showing us before. And we developed an algorithm that we call Prodigy. The aims to rank the mutated gene individual. We use the mutation data and the gene expression data and the global network like the previous study did, but we also look at separate pathways. So pathways are more focused, small group of interacting genes that are involved in a particular process in the cell. So the reasoning was that if a mutation affects a pathway, these are genes that are in the close neighborhood to it, and we can follow the changes along the chain from the driver to the pathway, and and the impact on changes on the net is not just random distribution in the network, but it will be focused in pathways. So here is the rationale. Suppose this is one pathway, and we have a mutated gene, and there are genes that are differentially expressed. They were different in the patient compared to normal. So these red guys are genes that were changed. So if this is indeed the driver, somehow the signal needed to propagate from the green guy to some of the red guys. But we want to limit this propagation, so we give a prize for reaching every red node, but we penalize for using the edges and the intermediate nodes that are not changed. And the penalty of the edges depends on how confident we are about the interaction, and the penalty on the intermediate node depends on the degree of the nodes. Nodes of high degree will, just by default, propagate the signal to many, so we want to downgrade their signal, so there is this parameter alpha of penalizing for the node degree. Now, mathematically, this can be put into the problem of a prize-collecting Steiner tree problem. So we have a root, and we have a penalty for every edge, and prize or penalty for every node, and we want to find a tree rooted at the root of maximum weight. Okay, so you see that in this example, perhaps this was the solution. We didn't reach this red node because the price of reaching it was higher than the penalty we would get from it. Now, this is an NP-hard problem, but there are good heuristics available, and actually we used an algorithm developed in Ernst Frankel group at MIT, who has been using this model for related problems. And we tested it on 5 types of cancer data, for about 2,500. And we used several resources of pathway databases. How did we evaluate our performance? We compared ourselves to the 2 previous algorithms, and we also compared ourselves to centrality measure that just measure what is how central a node is in the graph. So I won't get into the definition. These are classical definitions: degree, closeness, and betweenness. They just measure how central a node is. And what is how can we test our results? So we use this database of driver genes. I should emphasize that these are general drivers genes. They are not personalized. There's no data available per individual sample on the drivers. So this is the best we could do, and this is also what the previous studies did. Okay, so here are the results. You see here the F1 score, so the average of the precision and recall for the 3 algorithms. And as you see, this is the ranking of the top genes. So this is the top, and this is going down. And you see that Prodigy is doing substantially better than the previous methods. We also so we repeated the result on the 3 databases and got similar results. What is interesting to see here is that the centrality measures are actually performing better than the previous published algorithms. So if you just ignore the expression data completely and just look at how central a node is in the network, you get better results than by using these algorithms. So this is a word of caution which is known in the community. There are those high-degree proteins or genes called hubs, and they must be accounted for in every study of this type. One interesting question is that we ask, since we are collecting information across multiple pathways, so we collect the score of the prize collecting Steiner tree on each pathway separately. There could be a few hundreds of them. So how important is this collection? And as you see, for each of the cancer types, by using more pathways, the performance increases. And depending on the cohort, there could be more or less pathways that are needed. And so this approach of collecting information across pathways is highly powerful and improves over what was previously done. So to summarize, we are now on the verge of a new era of precision medicine. There's a tremendous amount of work to be done, and here I gave 3 examples from the type of work that we've been doing, showing that recommending the treatment to high blood pressure could be improved by losing the longitudinal data, developing a new, simple, and better multi-omic clustering algorithm and applying it to many cancer types. And finally, Prodigy, a new algorithm for prioritizing cancer driver genes in an individual patient. Thank you very much, and I thank my group.
We have time for a few questions. Moshe?
I thought you would ask, where is a sharp P here? Yeah. Oh, yeah. So I should have warned in advance that complexity is way hidden here. And
there was NP-hard once.
Well, okay, yeah, there was, yeah. But we use the heuristic. What can we do?
Sure. Yeah, Amir.
Amir.
From my experience,
from my experience, biological models tend to be very unstable. Do you have the same impression, and what are the possible ways to stabilize, to make models more significant for biological research? I mean, what I mean is sometimes you change the small parameters and the whole model stops working correctly.
So I think it's less related to the work we did here, but we had another study where we where we looked at a very large set of multi-disease data set, and we evaluated how good the prediction is as the data set grows. And so I should say that to follow up on what you said, there is a classical example of the predictor for breast cancer. So about 15 years ago, a Dutch research group developed a predictor for recurrence of breast cancer in women based on the biopsies, and it turned out that it was very accurate but extremely non-robust. So the prediction was good, but the set of genes would change dramatically if you just tweak the data. I don't know, remove 5 patients or remove 20 genes. So on one hand, you could say, you know, so we did a follow-up study on this larger cohort, and we showed that if you increase the data set, the stability of the predictor increases, but very slowly. So even with the 25,000, we could reach 25,000 patients. We could only reach Jacquard score, which measures the overlap in the gene set of something like 50%. So it will improve. It will improve very slowly. We need larger cohorts. And as I said, there are 2 aspects to it. One is the quality of the prediction, and other is the stability of the predictor. So the model may change with more data, but if the prediction is relatively stable, the point is that this breast cancer study ended up as a test that is being applied now to hundreds of thousands of women, even in spite of all these valid caveats about the stability.